Teachers, I think Yuta is teaching a lot of them. He does a great job. All right, I have a lot of announcements to get through. The first one is registering for 102. All right, there are a lot of students that need to register, and we want to assure everybody who wants to take 102 will get in. All right, even if it doesn't look like there's a course available at your time at registration, we will make it happen. All right, so don't panic. You will get into a class whether you see that it's immediately available or not. One of the things that we've done to kind of help eliminate any of this confusion from happening is adding in a limbo section. All right, so if you are looking at classes and everything's filled up or the sections left do not fit with your academic schedule, you can register for a limbo section, and that will allow you to register for a lab. All right, because you have to be registered for the lecture before you can register for the lab. All right, now this is only academic conflicts. All right, it does not include sports, it does not include music, it does not include any other extracurricular activity. All right, but at least let you get your foot in the door, and we will figure out the rest from there on out. Yuta is taking care of that, and he is great at making things happen. Any questions about that? All right, and some other announcements. So the study guide is up and on the Moodle site. Now again, because I'm posting it ahead of time, there might be topics that we might not get to, and if it's on the study guide, I'll try to remember to remove it, but like last time, I did not remember to remove them, so there are some up there that you're like, we didn't cover this. If we didn't cover it, no worries. I also included a document of supplemental materials on the Moodle site. So I put it all the way down to the week that the exam is. It's the week of the 14th. All right, the supplemental materials is just a bunch of supplemental materials that I have posted up there for your viewing pleasure. All right, so instead of putting this at the end of every lecture, I'm just going to keep adding on to this for exam 3. All right. Now on that note, I am giving you an assignment that has to do with supplemental materials. All right, so this is a 2-point assignment, and you'll find it on Moodle here. All right, resource assignment. If you click that link, it's going to take you to this Excel sheet. All right, so what I want you to do is I want to crowdsource the supplemental materials. All right, I can find supplemental materials, no problem. All right, but I think it's going to benefit you to find your own supplemental materials. And so if you go to the bottom there, there's another tab. All right, it's going to tell you what topic you're assigned and what date that's due. All right, so not everything's due at the same date, so pay attention to when this is due. If you have a conflict with the due dates at all, let me know right away, and we'll make sure we fix it, make it work. All right, so everybody has access to this here. It's worth 2 points, and I am looking for resources that are unique. All right, so if something's already been posted on your topic from another student, make sure you find something else. All right, if you post something that's a duplicate, I'm only going to be able to give you half credit, and I can go in, everything is time-stamped, and see who posted what first. All right, so if you try to delete somebody else's, I will know about it. The resources might be a video, they might be a reading, it can be anything that the public has access to. So if you need to sign up for something, all right, that immediately eliminates it. All right, so I know TikTok, I am not on TikTok, I don't know about TikTok. But I know you have to create an account. I don't know if you can watch it without creating an account. So if I can't watch it without creating an account, that right there is going to be a resource. as long as I have access to see it, all right? So maybe it's posted on another website. I don't have to sign in to view it. That'd be quite fine. Then I put in there to use your textbook as a resource to help guide for your resource hunt. All right, so if you remember on the schedule, every week it tells you what chapters we will be looking at. So if you are assigned Mendelian inheritance, all right, these are the chapters you can look at to get an idea of what you should be searching for. All right? Does that make sense to everybody? Sounds good. Yes. You only need one. Yep. So I put an example up here of the mitosis topic. It's actually mitosis and meiosis. But I put my name, the topic, short description, just so it's not a bunch of random links, and then the link to the resource. All right. And then the last announcement is that assignment 3 is ready. It is online and ready for you to complete. It is having you listen to a 30-minute podcast and answering some questions based off of that. Right? It should be a fairly easy assignment. It's worth 5 points. It's due November 15th, so you do have quite a bit of time to complete it. You could complete it by the end of the day today. All right? There's nothing that you need to wait on class material to complete this. All right, any other questions before we move on? Awesome. All right, so we're going to start with meiosis today. And the first thing I want to do is review mitosis. All right, so it helps to know mitosis before we go into meiosis. Now, I feel like I might have been off my game on Friday's lecture, so I want to just go back and review this, make sure we're all on the same page. Answer the questions of what steps of mitosis are represented in these circled cells. And then I want you to come up with your own mnemonic for remembering the steps of mitosis. All right, so mnemonic being when we learned oxidation and reduction, we used the mnemonic oil rig. Oxidation is losing, reduction is gaining. All right, so come up with your own unique mnemonic. All right, and then if you do need a review of mitosis, there's a link to a nice video that's going to help go through step by step again. All right, have at it. This should take a few minutes. Feel free to elaborate and talk to them. Yes. For 1 through 6, I want you to identify in those specific cells what's happening, and then you want to put the mnemonic for the whole process of mitosis. Oh no! Don't worry.
I'm never gonna forget that now.
Any questions since I'm here?
I'm never gonna forget it. I'm never gonna forget that. Okay.
In my AP bio class, just those they were cells, but they were like all dead cells, so not like my bad.
You can read it. But it's just a right topic.
You guys want to see my notes from yesterday? No, sure. Anna,
It's just like way too
Anna, have you seen my notes before? Okay.
You get a chance to come up with a mnemonic to answer the questions.
Yeah, but I can't say it out loud.
All right. I mean, if
But
it
it
helps you
really
remember,
helps. Really helps.
that's
I don't
good.
think we'll ever forget it.
That's what counts.
Ready?
That's going good on the
No, stop.
Wow. Try to gauge your mom's at.
Okay, question.
Yeah.
Is it okay to listen to Christmas carols today? That's like, that's our discussion.
Oh gosh, it depends on the mood I'm in and how my answer's gonna differ.
Okay.
Most of the time I'm gonna say not till after Thanksgiving.
I respect that. See, I respect that.
But sometimes it's the Christmas season.
Even when it starts to snow, like I'm like, I want it.
Yes, yes.
These are my new theory notes. And I'm telling you.
Oh, okay. Wait.
Oh, me too. But she hears it. She's like, I had books too, but I always forgot it.
Oh, no, she's good.
She would try to.
I'd be like,
It's not really. Where did you start? Yeah, but she got. Oh, yeah.
All right, anybody need more time to come up with a mnemonic?
Mnemonics are my secret talent.
All right, nobody's saying yes or raising their hand, so I'm gonna assume you have one. Who wants to share?
no.
I don't think you want to hear mine.
It's not. Say that word.
I mean, it is a college class. Whatever you feel is
Oh,
safe
no,
for
it's,
you.
it's pretty bad.
But
It's very.
all right.
Yeah. It's explicit.
Anybody else have a mnemonic they'd like to share? Yeah.
I said I prayed prior to meeting after Tom Cruise, but the first that doesn't stand for anything. But
It helps me remember it. All right, I like it. Very unique. You're gonna remember that. Anybody else have one? Yeah.
Isn't popcorn popping miraculously, awesomely, totally cool?
All right, here we go. I hear some very creative mnemonics here. I personally, I don't have a mnemonic that I came up with. I use the mnemonic, the PP mat, that you share online. That helps me remember it. So, all right. So, in terms of the cells we're looking at here, what might be going on in cell number 1? What phase of mitosis? Prophase, yeah, and why would that be?
things are starting to get condensed the DNA. It doesn't look like a blob, like a fingerprint.
Yeah, exactly. So things are being condensed. We can actually start to pick out where the chromosomes are. All right, and that happens in prophase. All right, what about number 2? Telophase, yeah, it's a telophase. It could be, depending on how the angle of the microscope here, it could be cytokinesis. It's kind of hard to tell with that one, but I want you to think about it. But essentially, we're seeing the formation of 2 individual cells. So that could be telophase if it's still one cell in one cytoplasm, one continuous cytoplasm, or it could be the beginnings of cytokinesis if that cytoplasm is starting to split. Now, it could also be that we just took an image of one cell on top of the other, all right? But it could also be these other processes. What about cell number 3? Metaphase. And why do we say that? They lined up. Yes, lining along that metaphase plate. All right, what about number 4? Anaphase, because they're separating apart. What about 5? Interphase. Yeah, not technically part of mitosis, but what's preceding mitosis here? Interphase. Nothing's condensed. We can't make out individual chromosomes. All right, and what about 6? I hear prometaphase. It could be. Yeah, we could have those spindles attached and they're starting to line up. It could also be. Any other guesses? Everyone said prometaphase? Wow, I'm surprised. All right, I would say this could also be considered metaphase because we're lining up along that metaphase plate. So it could be maybe we're at the end of prometaphase and the beginning of metaphase. All right, nice. I think that went nice and smooth. Everybody has a clear idea what mitosis is. So we can move on to meiosis. All right. So, talking about a multicellular organism, we have sexual reproduction, and that's going to involve a special kind of cell division, or meiosis, all right, because we have to form these gametes, egg and sperm. All right, so when we're talking about meiosis, ploidy is going to be an important key to know, or key term to know. All right, so ploidy is the number of each type of chromosome present. All right, so in the top here we have a, say there's a cell around these chromosomes. This cell has a ploidy of essentially 1, all right, it's haploid, all right? There's 1 of each type of chromosome, all right, versus diploid. Now we have duplicates of each chromosome, so we have 2 of each type of chromosome. So its ploidy would be 2, or diploid. Right, now we can also have organisms that are triploid. All right, of each type of chromosome, we have 3 replicates. All right, another thing that can end up being confusing when we talk about chromosomes and replication is if we look at this diploid example here, all right, we have pairs of chromosomes, right, but we have 6 chromosomes total. All right, so we could say in this cell here, see if I have a piece of chalk, we could say 2n equals 6. All right, we could say 2n because it's diploid, and that's how we designate diploid organisms. And we could say 6 because it has 6 total chromosomes. All right, now these chromosomes are paired up, so they're not all individually unique. But we know because they're diploid, all right, we have 2 chromosomes of each type. All right, is that making sense? All right. So this becomes important when we start to look at life cycles. All right, so in our human life cycle here, we have part of our life cycle that is haploid and part that's diploid. All right, now for us it's mostly diploid, but in mitosis we start with a diploid cell and we end up with replicates of that cell that are also diploid. All right, diploid to diploid. But in meiosis, we start with a diploid cell, all right, and we end up with these gametes that are haploid, right? They have half the genetic material. All right, and we can see here n equals 23, n meaning that it's haploid, 23 has 23 chromosomes. The zygote here, that's beginning of human development, right? 2n equals 46. 2n, we know it's diploid, and we know it has 46 chromosomes. Now, a good way to remember how many types of chromosomes there are, we can go 46 divided by 2, we're going to get 23. 23 different types of chromosomes. All right, so life cycles. Hugely important when we're talking about sexual reproduction. All right, it's a sequence of events that's going to produce another generation of organisms. And depending on what organism we're talking about, they're going to have different types of life cycles. All right, so it might be a diploid dominant species, haploid dominant, or in terms of plants, they might have alternations of generations. All right, so let's look at a diploid dominant species. We're a diploid dominant species. Mice are, most animals are. We spend most of our time as a diploid organism. All right, so for example, looking at this cell cycle on the bottom here, life cycle, we have our diploid zygote that's going to go through mitosis and produce a whole organism, in this case a mouse. All right, that whole process, we're talking about diploid cells. The only time we're talking about haploid cells are when we're talking about sperm or an egg. All right, so very little, a percentage of our cells are actually haploid. We are a diploid dominant species. Right, now that's different from a haploid dominant species. So this is very common in fungus. So the mushroom you see growing out of the ground is going to be haploid dominant. Right, it spends most of its lifetime as a haploid multicellular organism. So all these cells are haploid. The only time it becomes diploid is when it creates a zygote. Right, so when the reproductive cells come together and create a zygote, that is when it is diploid. Now this zygote goes on to immediately go through meiosis and create haploid cells. Right, so it's this one little slice of time in this life cycle that is diploid. Now this is different from most plants. All right, most plants have an alternation of generations. They spend more or less equal time as being haploid and being diploid. Right, so the plants that we typically see outside are going to be the sporophytes. They are diploid. These sporophytes are going to produce haploid spores that are going to grow into a multicellular organism, typically called the gametophyte. All right, those are all haploid, the haploid multicellular organism. And then eventually we'll get these haploids, a sperm or an egg, create a zygote, and the process starts all over. All right, so think about equal time spent in each. All right, so when we're talking about meiosis here, we're typically going to be talking about a diploid species, and we're typically going to be talking about humans. All right, humans have 46 chromosomes, all right, and 23 chromosome pairs. Right, now chromosome number is going to vary among species. Right, if we're talking about a dog, they have 78 chromosomes versus a fruit fly that only has 8. Right, so this is pretty common to have variation among different types of species. All right, so let's shift our focus to meiosis. How exactly are we going to get these haploid cells? Right, we're a diploid dominant species. We need to produce some haploid cells in order to create another organism. All right, and once we have these two haploid gametes, we can create a zygote, which will go on to go through mitosis, create another version of us, essentially. All right, so meiosis is that process by which these haploid cells are produced. All right, so for example, the example given here, we have a diploid human cell, contains 46 chromosomes and 23 pairs. All right, but the human gamete, it's haploid, so it only contains 23 chromosomes. There's no pairing going on. All right, so meiosis is split into 2 stages, essentially, meiosis 1 and meiosis 2. Now, they're very similar, but there are some differences between them. And the stages of meiosis 1 and meiosis 2 are also very similar to mitosis. All right, so just like mitosis, a cell going through meiosis 1 is going to begin by going through the G1, the S, and the G2 phase. All right, they're going to go through interphase. Now, during meiosis 1, the diploid parent cell is going to produce 2 haploid daughter cells. All right, and we're going to separate out homologous chromosomes from each other. All right, what is a homologous chromosome? Does anybody remember? Yeah.
Chromosomes that have show the same genes, so both show eye color.
Yeah, they have the same genes on them. So think of humans, we have 23 pairs of chromosomes. All right, let's take chromosome 1. We have 2 chromosomes in that pair. Each of those chromosomes are going to be homologous. They're going to code for the same type of genetic material. All right, so at the end of meiosis 1, these haploid cells are going to have no pairs of homologous chromosomes. So let's go through meiosis 1. And we're going to spend more time on meiosis 1 than meiosis 2 because this is where most of the differences occur. All right, and we're going to start with prophase 1. So in prophase 1, chromosomes are going to condense, all right, and we're going to form bivalents, all right, in a process called synapsis. And this is where crossing over can occur and where we can get the exchange of genetic material between chromosomes. So what is a bivalent? All right. I'm going to go over here and actually draw it out. I think that's going to make more sense. All right, so say we have a diploid organism. And chalk decided to become diploid as well. We have our diploid organism here. Right? They're going to create sister chromatids because they're going to duplicate. Right? So after duplication, we form these sister chromatids. All right, so we essentially have 4 copies of the same type of genetic material. All right, we have 2 homologous chromosomes turning into 2 sister chromatids. All right, at this point, we can consider this a chromosome, and each individual side a chromatid. Right, and I'm sure you're going to go over this a little bit more in depth in lab. Lab this week is focused all on mitosis and meiosis. Now, a bivalent is essentially we're going to put these 2 together. All right, I'm going to do my best to draw it here. Let's see. All right, so basically we have one on top of the other. We're forming a bivalent. Sometimes you will see this called a tetrad. I think in your lab manual, we're calling this a tetrad instead of a bivalent. Means the same thing. All right, so once we have a bivalent, all right, crossing over can occur. And this is where segments of chromosomes are going to exchange with each other. So if we look at crossing over, all right, crossing over can occur at many locations along our chromosomes and on both sets of non-sister chromatids. All right, and essentially what we're doing is we're swapping genetic material. So if we look at our homologous pairs here, all right, so we have 2 sets of sister chromatids. We can say one came from mom, one came from dad. All right, essentially we're going to break it apart and swap it between the 2. All right, so we're creating some genetic diversity here. And I put another image on here just to give you a clearer picture of what's going on. We're not going to go into all the nitty-gritty details here, but essentially we have a bivalent or a tetrad. All right, the area where crossing over occurs is called a chiasma. All right, so if we look at our tetrad, we have the breaking of homologous chromatids. All right, so there's a break there. A chiasma is formed, and we're essentially going to swap the genetic material between the 2 sister chromatids. All right, the end result after meiosis 1 and meiosis 2 is that we're going to get cells with variation of the chromosome that was taken part of crossing over. All right, so this is one step that we're going to get variation in meiosis. Does that make sense to everybody? All right. So we've gone through prophase. Crossing over has occurred. Chromosomes are condensed. The next step is prometaphase 1. All right, now a quick side note on this is some textbooks will not consider meiosis to have prometaphase. All right, and in your lab manual says that prometaphase does not occur. There's only early prophase and late prophase. But your textbooks considers it a step, so we're going to talk about it as a step so that it follows along with your textbook. All right, prometaphase is kind of the in-between step. The spindle apparatus is complete. All right, we completed our spindle apparatus. And the chromatids are going to be attached to the kinetochore microtubules. All right, so we're attaching our microtubules to our sister chromatids. Now the difference between meiosis 1 and mitosis is that we're attaching pairs of sister chromatids to a single pole. All right, so if we look up here, we have these pairs that are grouped together. Each side of the pair is attached to one pole, right? One or the other. That's different than mitosis. Right? In mitosis, they do not line up together. Basically, we're splitting apart sister chromatids in mitosis. All right? Meiosis 1, we're not splitting apart the sister chromatids. We're just essentially splitting apart the bivalents. All right, metaphase 1, the bivalents are going to be organized along that metaphase plate as a double row. All right, so the presence of a double row, that is how it's different than mitosis. All right, and we're arranging them randomly. All right, and this is where we get the term independent assortment. So we can arrange our bivalents like this possibility 1 here, all blues on one side, all reds on the other side, or it might randomly arrange to be opposite of that. All right, so blue, red, red, blue. All right, so in this way, we get more variation introduced. So if we look at those cells after it's gone through a complete set of meiosis 1 and mitosis 2, right, we see we have daughter cells with different combinations. All right, so this independent assortment is also introducing variation. All right, now there's a lot of variation that can occur. We're mostly looking at examples with 2 homologous chromosomes, or 2 chromosome pairs, excuse me. I think I even said that terminology wrong. We have 2 different types of chromosomes. All right, that's generally the example we're looking at. But in humans, we have 23 types of chromosomes. All right, so if we're a diploid species, we can estimate how many random variations we can have by going 2 to the power of n. All right, 2, and then we have n, which is the number of chromosomes per set. All right, and we have 23 chromosomes per set of our chromosomes. If we look at our karyotype, we have 23 sets. All right, that leaves us with over 8 million possibilities. All right, so there's a huge range for variation in independent assortment. All right, the last few steps of meiosis 1 occur with anaphase 1. Right, where we're separating the separation of homologs. All right, so those bivalents are separating apart. All right, and then they're going to migrate to each of the poles. Telophase is going to occur. The nuclear envelope is going to reappear. And then we have cytokinesis, and we're creating 2 cells. All right, and these 2 cells, they are now haploid. All right, they only contain 1 chromosome of a specific chromosome type. All right, so end of meiosis 1 happens fairly similar to meiosis or mitosis. Meiosis 2 happens even more similar to mitosis, with the exception being that after a cell has gone through meiosis 1, it's not going to go through interphase. It's just going to immediately go right into meiosis 2. All right, so there's no S phase, no interphase. The events in meiosis 2 are so identical to mitosis that we're not going to go over them step by step. All right, the biggest difference between meiosis 1 and meiosis 2 is how the chromatids are separated out. All right, so sister chromatids are separated in the anaphase step. All right, so we're looking at anaphase here. We're separating out the sister chromatids, just like we saw in mitosis. In meiosis 1, we were separating out bivalents. All right, so that's the biggest difference that occurs between meiosis 1 and meiosis 2. All right, so I talked about how we can see variability, and I just wanted to recap on that topic here. First of all, we have genetic material from 2 individuals. All right, each parent contains a different set of genetic material. Same type of genetic material, but might encode for different things. Blonde hair versus black hair, for example. We have independent assortment of chromosomes during meiosis, right? How they're lining up on the metaphase plate during meiosis 1 specifically. And then we have recombination or crossing over during meiosis, specifically in prophase 1. All All right, so there's a huge amount of ways that we can have variation when we're talking about meiosis. All right, and I kind of went over a lot of the differences between mitosis and meiosis, but I just wanted to reiterate that mitosis, we're producing 2 diploid daughter cells. Meiosis, we're producing 4 haploid daughter cells. All right? And I want you to think about this a little bit more in depth. All right, I talked about a lot of them. Review what I talked about, maybe come up with some that I didn't talk about. List some of the differences between mitosis and meiosis. All right, and work together. Oh yes, it always is. Yeah, it's going to be history, biology, and I'm doing this after. Yeah. It's fine.
I think he didn't move him. He lives in Kauai, but he's literally only been okay. I also supposed to be twice, but I don't even know how to like grade.
Yeah. So what are they?
Can you show me how to do it on here?
I thought this was the bus. Yeah. And then this is I don't know what I'm confused.
Yeah, so it's looking at
I'm not excited.
yes, yes. Altogether, active replication occurred in a diploid cell. One to the end, then it's going to be yes, haploid.
You roast.
Meiosis, yes, yes. Yep. So you can say that the original diploid cell. At the end, I know, yeah.
feel this one bring it together. I'm sorry. It's just I want to see more of what's 2 by 1? Organic.
Well, I'll show you what I have.
I mean, honestly, I might take Kelp back home for the summer.
Because I think these, like I think this idea is 244 beats.
I have a question.
Yeah.
There's steel probably in the sugar bio, but
Yeah.
what's the difference between considering one, two?
Wait, yeah, why does she know?
One's Genesis and
We have those classes.
There you go. All right. So, what are some of those differences between mitosis and meiosis? There's quite a few of them. Yeah.
In meiosis, you're changing the chromosomes or whatever, but in mitosis, you're replicating them.
Yeah. So talking about ploidy. So in mitosis, we're going from a diploid to a diploid, and meiosis, we're going diploid to haploid. All right. That's one major difference. What's another? There's a pretty obvious one. We have mitosis. We have meiosis. That's split into 2. Yes, 2 stages for meiosis. All right. What are some other differences? Oh, I thought that was yes.
In meiosis, there's crossing over and like separating triads.
Yeah, so in meiosis, we have crossing over that occurs. We also have separation of triads. All right. So in mitosis, we don't get those bivalents. Anything else anyone would like to throw out? All right, there's a lot of them. All right, and this table, I believe, is out of your book here. This table goes through a lot of the comparisons between mitosis and meiosis 1 and meiosis 2. Now, in your slides there, I put this table right at the end. All right, so this table is in your lecture slides. All right, I'm not going to go through all these, but do give it a look over. It helps you kind of understand what's going on. All right, I'm immediately going to go into another group activity, and this is going to involve a lot of drawing. So I'm going to pass around some paper if you'd like paper, if you don't have notebook paper. But I want you to draw out these cells. All right, so let's go through this first cell together. If I say a cell is 2n equals 6, is 2n meaning diploid or haploid? Diploid. And what does the 6 mean? We have 6 total chromosomes. Yeah, so if we take 6 divided by 2, we get 3. So we know we have 3 pairs of those chromosomes. All right. Awesome. Go ahead and go through this. Let's see. I think most of you have notebook paper, but raise your hand if you want another sheet of paper.
41.2.
Okay, if you want to pass that
Are
one
we
down.
turning this in?
You're not turning this in. No, this is just for your own use.
You want to see what Emma just texted me? Sorry. You want to see what
Oh gosh. All right, everyone has paper that wants paper. Awesome. Alright, I'm gonna assume you're gonna ace this part.
Yeah, you got you will. I actually do. Okay, that's good. How about teacher? This is like a conversation starter for me.
Yeah.
Yeah. So when we say 20 plus 6, this is the fourth duplication. Alright, so we have, so essentially, the four duplication, this would be 1, this would be 2. What you guys started is yeah, exactly. Yep,
How's this looking?
How's it looking? Let's see. It's looking good.
Do you think we should like color code like it when it crosses over?
If you feel so inclined, that'd be awesome.
Do it.
All right, we're not going to have a chance to go over this in class today, but this is where we're going to start up on Wednesday. Right? So on Wednesday, we will review this group activity. Homework, there is a video I'd like you to watch for Wednesday. Alright, so make sure you watch that video, and then we have a lot of upcoming assignments just for you to be aware of.
Nothing.